My name is Travis Glenn. I am a faculty member here at the University of Georgia. And just to give you an idea, hopefully I can make the slides advance. There we go. I am a farm kid from the Midwest. Did my degrees at Iowa State University, the University of Michigan, PhD in zoology at the University of Maryland, where I worked at the Smithsonian on old dead stuff. And then I went to a postdoc at the University of South Carolina. Then I joined the University of Georgia at the Savannah River Ecology Lab, and I was there for more than 9 years before I moved up to the mothership here in Athens for UGA. And so I've been here in the Department of Environmental Health Science since 2008. And I am a professor and currently serving as head of the Department of Environmental Health Science. It is part of the College of Public Health here at UGA. And I also serve as the director of the Institute of Bioinformatics here at UGA. Okay, so that's a little bit of background about me. And if I ever, cover information that you're wondering, wait, can you clarify? Feel free to just chime in, and you can stop me. You can also put information in the chat and I'll try and circle to that. If you know me at all, I like to start with a little bit of trivia questions. What I want you to do is to type the information into chat, but don't hit enter until I say so. Okay, so the first question is, which came first, D-Day or publication of the experiments demonstrating that DNA was definitively the molecule of inheritance? Okay, so you can say D-Day or DNA. Okay, get ready, type in your answer. Da, 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 da. Okay, go. DNA, DNA, DNA. Okay, so that is correct, but only by a few months. It was February of 1944 when Avery et al. Was published. Okay, so if you said D-Day, you were not off by much. All right, next question. Which came first, Sanger DNA sequencing or Star Wars? And actually, there were 3 different kinds of sequencing described all in the same year as Sanger. So type your answer. So DNA sequencing or Star Wars? Okay, go ahead. Enter, enter, enter. Okay, Star Wars is correct, but again, only by a few months, at least for Sanger. So Sanger came out, was published in December of 1977. Star Wars, the first one, came out in that summer. Okay, next one. Which came first, 9/11 or the first draft of the human genome sequence? Okay, so type your answers, but don't hit send. Okay, well, go ahead, hit send. Go, 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 go. What do you think? Okay, and the answer to that one is first draft of the human genome sequence. Okay, and that was announced in 2000, published in February of 2001. 9/11 was in September of 2001. Okay. Which came first? Sequencing an individual DNA molecule or completion of the Human Genome Project? This one's a little tougher. So, DNA or genome? All right, you ready? Go ahead. All right. And this one is a little complex. It's actually sequencing individual DNA molecules came first. So the completion of the Human Genome Project was announced in April of 2003. The sequencing that became PacBio sequencing was published in Science in January of 2003. Okay, so last trivia question. Whoops. Which came first? Completion, and I mean telomere to telomere, no gaps, human genome sequence, or the January 6th Capitol riots? Okay. It's another head scratcher. All right. Go ahead. Lots and lots of guessing. Okay, whoops. And the answer to that one is the Capitol riot. It was 2001. The telomere to telomere T2T sequence was in 2022. So by more than a year. Okay. So just a little bit of background. And again, I kind of try and put these together to give you some major events, to give you an idea of when these things occurred, right? And so the idea of number one is we haven't known even for 100 years that DNA was the molecule of inheritance, okay? We haven't known longer than Star Wars has been around that we've been able to sequence DNA, okay? So just keep in mind, and it's really, in the past 20, 25 years that we really could sequence at scale. Okay. So what we're going to do now is talk about how can we use all of the advances and tools? What are they? What's the current state of play? Right. And in particular, the goal of doing, digging into all these details is to understand how can we get DNA data efficiently so that we can answer interesting biological questions and solve problems in public health, right? That's really the goal here. Okay, so in general, there's probably three goals that people have when they're thinking about how they're going to acquire data in general, right? And this applies also for DNA sequencing. So in the chat, what I'd like you to do is to rank what's most important, speed, accuracy, or cost. Okay, and there's no right or wrong answer. But if you had to rank them, what would you rank first? Or if you all three, how would you rank the three? Okay, lots and lots of people, accuracy first. Cost second, speed third. That seems to be what I'm seeing a lot of. Okay, great. So accuracy. How do you know if a DNA sequence is accurate? So one of the things that people will do is they'll talk about quality scores or Q scores. They also might refer to these as Fred-like scores. And so when we talk about accuracy for DNA sequencing, it's on a log scale, and the first digit tells you how many zeros there are or how many nines there are in a percent basis for incorrect calls or accuracy. Okay, so if you have a quality score of 10, you're 90% accurate, which means you have a 10% probability of being wrong. Have a Q score of 20, you're 99% accurate. 30, 99.9% accurate. Okay, so Q30 is really quite good. And this is a number that people will use as sort of a touch point for a lot of the things that we're going to talk about. And that's, again, it's 99.9% accurate. Q20, 99% accurate. Okay, and if you keep on going, it just gets smaller and smaller, your probability of being wrong. Okay. When we talk about assays, there's really trade-offs though between good, which is, accurate, fast, how quick can I do it, and how cheap is it, right? And it's pretty difficult to do all three simultaneously. And in fact, some people will tell you only the unicorn is the one that can give you everything that you really want. Okay? So what we've really got to do is, can we find unicorns? It'd be great if we can, but if we can't, what are we willing to trade off? Okay. And in particular, How accurate is accurate enough? Is Q20 good enough? Or do I really need to get to Q30 or Q40? How fast is fast enough? Is 12 hours good enough? Does it have to be overnight? Is 24 hours? Where really am I okay with fast? And similarly with cheap. When does it become so cheap that the cost doesn't really matter that much per sample? Okay. And so we're going to think about all of those measures. All right. So again, cheap. How can I get the assays? Cheap, good. Is it accurate enough that I make the right call for my biological answer and therefore a public health recommendation? Is it good enough? And how fast? How fast is it? Okay. So we're going to make trade-offs, but again, our goal is to find the unicorn. Okay. And I'm going to show you how to get into the unicorn zone. But what's what's the goal? Okay. Again, in public health, a lot of the goal is to sift through a whole bunch of stuff and find the bad actor. How can I find my pathogen? How can I find the thing that is causing a bad public health outcome? Okay. Not only can I find it, can I characterize it? How is this actor, better or worse or different than a bad actor that I'm very familiar with? Who is it close to? Who is it similar to? Right? Or do I just have a whole bad community and I need to understand who's the most common, who's the least common in that group? Okay. And more specifically in public health, what I might do is go sample different areas, and then I'm going to try and find these bad pathogens. And so this is a slide that Megan Beaudry, a former student of mine, developed that described a project that we were working on with CDC. And so what we did in that project was to go and sample microbiomes, right? The surface microbiome in a bunch of different environments, extract the DNA. There might be, some particular targets that we're looking for, and then how do we deal with those targets, right? We can do metagenomics where I sequence everything. I can enrich for the bad actors that I'm particularly interested in, or I can do very specific assays like qPCR that target very well known and well characterized bad actors. And all of those assays are possible with NextGen. And it's really a matter of what are you trying to do and, what's the goal of the project that you're trying to do. If you're characterizing specific bad actors, qPCR will always be the most precise and it's the most sensitive assay, right? It's going to detect the bad actors in the fewest amount. Metagenomics, on the other hand, where I just sequence everything, is going to give me the most comprehensive view. And then there are some techniques that in particular next week's speaker is going to talk about is how do you enrich for bad actors so you're more likely to find the ones that are at low prevalence. Okay, but the key to both of these lower nuggets is doing next-gen sequencing. Okay, so I've used the term next-gen sequencing versus what? What was previous gen sequencing? So first generation sequencing was mostly Sanger sequencing, which I talked about, invented in 1977 by Thomas Sanger. And people will have done that now on capillary instruments for almost 30 years. And so people will use those terms interchangeably, Sanger sequencing or capillary sequencing. And the nugget about that is that it is one tube for one locus for one sample. Okay. And so a single researcher typically would do PCR on a sample, and then they would do sequencing on that PCR product. And a single researcher could sort of do a plate or a couple of plates per week. Okay. Reads are reasonably long, sort of 600-ish of good quality, sometimes longer if you're willing to accept lower quality. And because these capillary sequencers can do lots of samples, whole 96 well plates all at the same time, you've got to have some ability to handle that data and deal with it on a computer. Okay, how does that contrast with massively parallel or next-gen sequencing? Okay, the biggest nugget is that next-gen sequencing or massively parallel sequencing allows you to get millions of sequences per tube, not just one, right? Not the consensus of millions of molecules that all need to report the same thing. You can have a very heterogeneous sample and get millions of different sequences out of it. Each one corresponds to an individual DNA molecule in your sample. Okay, and whether the sequencing instrument multiplies that single molecule so it has enough signal to detect it, that's second generation, or whether it reads that individual molecule directly, that's third generation. That will vary among the platforms, and we'll talk about that in a few minutes. Okay, but by having these next-gen sequencers, that allows a single researcher to, deal with dozens of samples, up to hundreds of samples per week. Okay, the reads can be very short, depending on the platform, or they can be really long, hundreds of thousands of base pairs long. And it depends on the platform, and we'll talk about what those various platforms are. Okay. And of course, if you're generating that amount of data, it is critical that you have good computational systems set up to deal with that amount of data. Okay. And so, when I got into the sequencing game, almost 30 years ago, I never really imagined that it would be possible during my working career that we would be able to sequence full genomes, not just of humans, but of almost any creature on Earth that you were interested in. Okay. And so, like magic, next-gen sequencing makes unimaginable quantities of data a reality. Okay. So this is sort of the magic of my professional lifetime that has really, opened up the possibilities of what it's feasible to do. So now what I'm going to do is go through and talk about some of the instruments quite specifically, but I'm going to start with some overview so you get a feel for what the instruments are, what are the positives, what are the negatives. Okay. In general, there are 4 different sequencing companies or platforms that are well established. They've been selling instruments for years and years, and lots of people have them. Okay. You may or may not have them in your own labs, but you could probably find someone who has instruments of these types pretty easily. Okay. Those 4 platforms are Illumina, which is the big dog. They have lots of different instruments. We'll talk about some of them. There's Oxford Nanopore, which is nanopore sequencing, individual molecule sequencing. There's PacBio, which is again individual molecule sequencing. And there's still Ion Torrent, which was sort of the second generation of a technology that really was the first to market in next-gen sequencing space. All right, so this slide summarizes something about each of those 4 platforms, right? So the first thing is that it tells you how is the sequencing done. So Illumina does sequencing by synthesis in a manner that's pretty similar to Sanger sequencing, except instead of getting the consensus of, many molecules, you march with each stop nucleotide, one nucleotide at a time. Okay. Ion Torrent does synthesis, pyrosequencing is how that works. PacBio also does synthesis of fluorescent DNTPs where every nucleotide is fluorescently labeled, but it's not a stop nucleotide. Okay. And these are looking at individual molecules. And then Oxford Nanopore scans the DNA as it flows through a nanopore. Okay. And again, I'm going to talk about each of these just in a little bit more detail, but not a whole lot.
Travis, we have a comment in the chat.
Okay. Oh, you have a Clear Labs. All right, sweet. And there are some other instruments that I'll also talk about after we go through these four. So, and I'll try and talk about these other instruments, and then we can talk about positives and negatives as we wrap up. But thanks for that. Okay, so amplification is sort of the difference between is something a second generation instrument or a third generation instrument. Okay, so all of these, again, they start with single molecules in your sample, and then if they're a second generation instrument, they clonally amplify those individual molecules and then sequence that amplified product. But again, it's clonally amplified, sort of like going from a single bacterium on a plate to a colony, or they don't do any amplification at all, and then they sequence those individual molecules directly. Okay. All right. So the first one that I'm going to talk about is maybe the one that gets used least in public health, which is pyrosequencing. This, again, was the first commercially developed platform with the 454s. There have been numerous pyrosequencing companies that have come along and don't exist anymore. There are only two that are still alive. The most commonly used one is the Ion Torrent from Thermo Fisher. It is mostly used for cancer diagnostics. They have a couple of different systems that are available. The really nice thing about these systems is that they are well tuned to go from a standardized sample to an answer with very little human intervention between those 2 points. Okay, and so again, this sort of builds on more than 20 years of tweaking the system. And so it works pretty well, but it does have some specific downsides, and, there's good reasons why it doesn't rule the market. It sort of is a niche system that works well, again, for cancer screening. Um, but one of the biggest issues with it is that it has problems with accuracy, and in particular homopolymer runs, so runs of A's or C's or T's or G's just all in a row. It can't deal with that very well. All right, so the next platform we're going to move to is Illumina. Illumina is the dominant sequence by synthesis platform. It's been around again for more than 20 years. Started with genome analyzers, then it went on to the HiSeq family. There are now many instruments. All of these that I'm showing on this slide are still available from Illumina. Okay, they go from a low number of reads, like 1 million reads per run, up to 25 billion reads per run. Okay, the 3 instruments or 4 instruments that I show on this top row here, these are sort of the new frontline instruments. That's, if anyone's buying instruments, it's generally those four. The other 4 instruments down below here are mostly legacy instruments that people may still be running for one reason or another. But in general, it is not economically viable. It doesn't make economic sense to continue running those instruments, except that it takes time to revalidate all of your protocols to go on to the new instruments. So You're probably going to see, you may well be involved with this notion of, developing your protocols and, sort of rejiggering them for the new instruments. I'm going to focus on 3 out of those 4 instruments to give you an idea of, again, these are sort of the 3 most commonly used instruments currently. Those are also, the ones that Illumina is still selling the most of. The MySeq in particular is, there's a new version of the MySeq. They don't have available, even though they tell you about all the different configurations, they are still only selling kits for the 5 million and 25 million clusters per run reagents. They do not offer those 2 yet, but supposedly they will. Okay. The NextSeq has been around a bit longer. They have lots of configurations. This is the one platform where they dropped costs dramatically in the past year or 2 years for a lot of these kits, but they're still really not economically viable for the most part, unless you're running PE 100, PE 300s on that 100 million flow cell. That's a pretty good bargain. Almost any other configuration, you're better off to either go down to the MySeq or up to the NovaSeq, right? And if you're running a NextSeq, it's not because it's economically the most cost efficient, it's because you have control of it. Because, right, you can control, exactly what you put on it, when you put on it, what information comes through, right? If you're looking for economies of scale, you would want to move up to the NovaSeq X and collaborate with other folks. Okay, so what does that mean? Is it cheap enough that I should really consider that? And so on this slide, what I'm giving you is what's the capacity of all of these different instruments? Again, there's a lot of instruments here. These slides are going to be available so you can go back to them if you want to. I'm not going to, kind of run through the details here. I'm going to point out the 2 instruments that are the most economical to run, which would be the MySeq i100, the new version of that. And again, they're only doing the 25 millions up to now, but they'll roll out the others as time comes along. And then the NovaSeq X, where you really kick into high throughput, you're going to be using sample indexing. So you can run lots of samples all at the same time and then divide those run costs among many, many samples. All right. So are there any questions, other questions that people have currently? I'm going to keep rolling, but I'll look back at the chat here in case you do. All right, so one of the big things that I want you to get, right, is that next gen is cheap per pound, right? So per million reads, but you have to buy the whole truckload of sequencing. You have to do a whole run. You can't do a partial run on most of these instruments. There are a few that you can, or you can do a short run, but I'll point those out specifically. For the most part, you put in a batch of samples and the machine runs until it's done. You can't run it one lane at a time. Some, instruments you can, but for the most part on Illumina, you cannot. Okay. So what's the cost advantage of doing this next gen stuff? Okay. In capillary sequencing land, if you wanted 1 billion base pairs of sequence, that's over $1 million. But the buy-in cost for one sample, one read from one sample, is really cheap. It's $5. If I compare that to the legacy MySeq, it's about $1,000 per gigabase pair, right? So it's 1,000 times cheaper to get a lot of sequence. But the minimum buy-in costs 100 times more. And if I go to the NovaSeq X Plus, Again, I get even more orders of magnitude cheaper, but the minimum buy-in cost is still pretty expensive. This is a 10B lane. They do now have a 1.5B lane, which is even cheaper, but not many people are running those. Okay, but you could if you wanted to, but then this cost is going to go up, the cost per gigabase pair. Okay, so again, this is brand new data. I literally was downloading the cost comparison information on reagent costs for Illumina instruments last night and this morning. So these are the reagent costs per million paired end reads at PE 150. So that's sort of the comparison point that I tried to make. The range is what's the kit. So the lowest cost would be for the highest capacity kit. So it has the highest purchase cost, but the lowest cost per million base pairs. Okay, so for example, going from 36 to 130, the 36 is for the 25 million uh, cluster kit. The $130 per million base pairs or per million reads is for the 5 million cluster kit. Okay, so you can pay as much as $300 per million reads at PE 150 or as little as less than $1. Okay, and that's all within Illumina. It's just a matter of choosing which instrument and which kit. So there's, almost 3.5 orders of magnitude of difference just within the Illumina platform. Okay, so it's pretty important that you think about what's the throughput that I need? How fast do I need it? Can I, batch with other things to reduce my per sample cost? Right? And it's trade-offs and, what works best for you. Okay, so what are the strengths of Illumina? They've been around for a long time. They have a huge percentage of the market, probably less and less as time goes on because there's more different instruments that are available that people are using. But there's just a great uh, quantity of information about how to run these things, how to make the samples work. The company enables people to really be creative about sort of doing your own thing within the general constraints of the platform. And there are lots and lots of people who have done that. And so maybe one of the biggest reasons is it's an industry standard, right? You can compare your results with almost anybody else. All right, so what are some alternatives? One is from PacBio. Their newest instrument is the Revio. It runs up to 4 flow cells at a time. Each run is approximately 1 day in length. It has 25 million wells. Not all of those wells are occupied, right? It's probably down around 50% now with the newest chemistry, which is the best it's ever been. The length of the molecules is between 1 and 25 kb for hi-fi sequencing, which we'll talk about. So hi-fi reads are a circular consensus, and I'll show you on the next slide how you turn a linear double-stranded DNA molecule into a circle. And so we're going to sequence round and around that thing. And if you sort of say, what's the default way to de novo sequence a brand new genome from some organism with a large genome, PacBio is sort of the default way to go. Oxford Nanopore is not far behind, but it's still probably 70-30 sort of a split, or 2/3, 1/3, 2/3 go into PacBio. Okay. And one of the major reasons that people will, go with PacBio first is that the informatics is a lot easier, right? Because you're starting with consistent sizes of reads and you have very high quality. Okay, let's see if we can go forward. Hello. Hello. Awesome. Have I died? I seem to be dead. That is just super. Oops. All right. Hold on. All right. My slides are back. Do I have everyone else back?
We're still just seeing trees.
Okay, sweet. I'm gonna try this. How long was I gone?
You weren't gone at all.
Oh, sweet. Interesting. Everything froze. So that's just great. Okay. So this is circular consensus. So you start with your linear DNA. You ligate on dumbbells sort of onto the end of each end of your DNA molecule, which turns it into a circle. And then the polymerase can go round and round that circle. And what that means is that you're going back and forth and back and forth across that same insert time and again. And so as long as your errors are random, you will reach a consensus that is accurate. Okay, and that's how hi-fi works, and that's why it's pretty good, especially if you don't know what to expect, right? So for a de novo sequence. What's the newest thing that PacBio has brought out this year? They've brought out Spark chemistry. And so the deal about Spark chemistry is that it allows a much lower input of DNA into the gold standard library prep. There are additional library preps that you can use even less DNA for, but sort of the gold standard now is down to 500 nanograms, which is a lot more achievable for many more projects. And it's also much more efficient at loading those libraries onto the flow cell. And that means you'll get more sequence out for the exact same number of dollars that it costs to run the sequencer. Okay, and that just means that it's getting ever more cost efficient. All right, so Revio has been around for a couple of years, and PacBio is going through iterations um, to make things cheaper and better. And you can expect that that will be true. All right, what's another major technology that's out there? Oxford Nanopore. Okay, and so for Oxford Nanopore, the trick is they're not synthesizing new DNA. They're taking the DNA that you have and pulling it through a nanopore, and then they're reading the bases as it goes through that nanopore. And they're generating an electrical signal. And it's unfortunately, the pore isn't small enough to read one nucleotide at a time. It reads groups of three. One of the good things that Oxford Nanopore has been able to do in the past just 2 or 3 years is that they've developed a longer nanopore that has 2 read points. And so that's what's enabled them to get up to a Q20 score. And once you reach Q20, then that sort of, allows lots and lots of different applications to become possible. It is also the case that Oxford Nanopore has a variety of different instruments that are available. These little guys are portable, they're field portable. You can take them out in the field. I'm sure, since you all are in public health labs, you're well aware of those. Okay, so again, the sequencers here are low cost, they're portable. The quality threshold has gotten up over Q20, and you can sort of expect them to keep iterating and getting better and better over time. The reads are really limited by the length of the molecules that you feed in. It's also a super positive is that you can do a read until, which means you run you load your sample onto the instrument and you run it until you detect your bad actor. Okay. Once you've detected it, some minimum number of times, then you can stop running it, wash off your flow cell, and then reload it with fresh samples if you want to. Okay, and so that's a way of getting data quicker and to sort of be economical about your use of these instruments. So there's sort of lots of places for people to innovate, but what I would say is, it's taken a while for Oxford Nanopore technology to get here, but it really is at a point where people can use it pretty routinely out of the box now. And there's lots of, opportunities to be creative in your use of their instruments. Okay, so this is a slide that came from, one of the brochures for Revio that sort of summarizes what's the difference between PacBio versus Illumina or other Illumina-like instruments and Oxford Nanopore, right? And you'll see that PacBio and Oxford Nanopore really are pretty similar. Um, the Nanopore sequencers are not quite as good at calling indels, and it is also the case that almost no one will point out to you that the errors that Oxford Nanopore makes are not random. They are systematic. And what that means is if you sequence the same molecule over and over and over again, you will become more and more convinced of the wrong answer. Okay? So it doesn't mean that you can't use Oxford Nanopore. You can get consistent answers, which is probably the most important, but the consistent answer may not be, what truth is. But as long as you can convert from what the consistent answer is that they give you to what you need to know, you're still okay. But it is something that people need to keep in mind and they don't talk about very much. Okay. It is also the case, yeah, that you can do Oxford, you can do real-time analysis while it's sequencing. Um, and that will depend on the compute power that you have available, because you can go to their highest throughput instruments and you have to have some pretty beefy computational systems set up to keep up with them in real time. And not everyone will invest in that. And that's okay. Again, it just depends on what your needs are. All right. So I have a bunch of pluses and minuses about PacBio versus Oxford Nanopore. There's some trade-offs. I would just say, keep those in mind. Again, these slides will be here for you to go through. Um, Similarly, Hi-Fi versus the Promethion, that's probably the most direct sort of apples to apples comparison for high throughput. All right, so what are some of the other platforms you may not have heard as much about? So one is Singular Genomics. They came out with their G4 to try and compete against Illumina, which wasn't a very good idea. They don't sell that anymore. Now they have the G4X, which is focused on spatial genomics. And so that's probably a pretty good idea. An instrument you may see and you now can get access to at commercial labs, as well as some of the highest throughput genomics labs, is an instrument, the UG100 from Ultima Genomics. And so this is the other pyrosequencing platform. They've done a number of sort of interesting things to make read quality of homopolymers much, much better, especially for short homopolymer reads. And they've done a bunch of sort of innovative things that bring the cost down for a full 30x human genome to under $100. So if you're looking to do really, really big projects on relatively short reads, this is sort of a platform to be, to keep your eye out on. It seems that they've made it over the hump of actually placing some of their instruments into commercial labs. So we'll see if they can continue on that track. MGI Tech has a variety of instruments. They're very similar to Illumina instruments. They get there in a somewhat different way. They use DNA nanoballs as their way of generating clusters. Um, this is a company that is in China. It used to be run by folks from BGI. They have had a bunch of patent fights with Illumina and won a bunch of money from Illumina a few years ago. Probably the biggest, risk with trying to use their instruments is that it is a Chinese company. It is a Chinese high-tech company, and the US government and Chinese government are not getting along. So I would expect in federally funded agencies that it probably would be prudent, to consider other options. Not for any particular technical reasons, but just because of the reality of the world we live in. All right, another instrument to potentially keep in mind are the Aviti line of instruments that are from Element Biosciences. These, again, sort of give Illumina-like reads, but they get there in a different chemistry that's sort of an interesting chemistry that has a variety of advantages uh, um, on sort of very specific technical things that I'm not going to get into. But the gist is you can get Illumina-like reads from an instrument that costs about the same as a NextSeq, but you get per gigabase pair costs that's similar to the NovaSeq X. So it's a little bit in between those two instruments. And again, this might be a place that you can, that could find a place in an ecosystem in public health labs. We have used it. It is also, the case that there are differences between Illumina versus Aviti. And so you would need to think carefully about some of those things, right? And Sam is pointing out, upload speeds, different specifications. It's not going to be a one-for-one trade with Illumina. But I can tell you, we have sent our Illumina libraries on Avitis. They perform very, very similarly. So it is a possibility. Again, I'm not an advocate for any one of these platforms. I don't own stock in them. You can do as you wish, right? I'm just trying to, make them available, make some information about those available. Sam, I don't know if you want to jump in and maybe talk about why you chose the instrument you did from Clear Genomics, Clear Cell,
Oh, Clear Labs.
Clear Labs.
I can say we did not choose the Aviti. We were looking at they will send you one to try out and play with, and then they come pick it up, which was nice, but we ended up not getting it for newborn screening.
Yep.
For Clear Labs, I think we just got money for it during COVID. We don't particularly use it a lot.
Yep.
Brenna might know more about what we're using it for. We had a run going it on this week, but it's not one of our like daily drivers.
Yep.
The Clear Labs is just a liquid handler and it runs off of it can run samples for Illumina iSeqs or ONT. They're pushing more towards the Illumina platforms now, and I hope we will be using it more for we have a couple of assays that are well suited to be done using the whole, basically it's a Hamilton NGS star,
Yep.
whatever. And then it is just, they've extended the programming to be able to load it onto an IC cartridge or load it onto an ONT. And yes, it was COVID money because we were having so many samples that people wanted sequenced with rapid turnaround, and that offered us a more end-to-end process without requiring people on the bench 24/7.
Yep. Awesome. Yeah. And so there's sort of a whole universe of additional liquid handling instruments. Um, that I didn't get into at all, but that's, you know, again, something that we could talk about if that made sense at some point. Because yeah, there's lots of pluses and minuses to the different instruments that are available, and they'll make a big difference to your day-to-day. You know, how much time does it take? How repeatable is the assay? You know, how long does it take to get the robot up and running? Yada, yada. Okay, so I think what I'm going to do is do the big wrap up. All right, so here's my, you know, the end of seminar quiz for you, right? What's the most common NGS platform? Again, type it in, but don't hit send quite yet. 5, 4, 3, 2, 1. Hit send. Yay. All right. So everybody got that. That's a good one. All right. All right. How does PacBio achieve high quality sequence read? Okay. Again, type it in, but don't hit send quite yet. 5, 4, 3, 2, 1. Go ahead. All right, circular consensus, hi-fi reads, right? So they're reading the same molecule over and over again. And again, ONT, one of their great innovations was to have 2 read points. Okay, and you can imagine they might at some point roll out 3, but it's sort of diminishing returns. 2 was a really big deal about getting up to Q20 scores on the read accuracy. All right, how can you identify or verify that your reads are high quality? How do you know that, you know, the answer you got is likely to be correct? Okay, again, type that in. And go. Red scores, exactly. Okay, Q scores, right? And the problem that ONT has, you know, or any platform that has a systematic bias to their errors, is how do you deal with that? Because Fred is not built for that. It is built on random errors. Okay, so just a little truth in advertising about ONT. You know, there's a little more work about dealing with the data you get from them. All right, so the last thing, you know, again, I'm all about, it's fine to use lots of different instruments. Those are great. I'm, you know, an exploratory kind of person, but I want you to be, you know, to understand the trade-offs, right? So if you take something like an aluminum iSeq and you do PE300s on an E. coli isolate, and you do whole genome shotgun sequencing, and then you try and assemble it. If you do that, your genome sequence has pretty few errors, but it's in hundreds of pieces because you can't get the repetitive elements to go together from short reads. Okay. On the other side, ONT, with a MinION, you can assemble everything into a single contig, which is great, but it's going to have quite a few errors in it. Okay, less and less as time goes on, but still substantially more than Illumina at this point in time. The alternative to that is PacBio Hi-Fi, which is one thing with very few errors, but it has the highest cost. Okay.